morning, students. Welcome to biochemistry lecture. In today's lecture, we are going to see another important topic in the carbohydrate unit, that is pentose phosphate pathway, triple P. Let's go into the topic now. It is an alternate route for the glucose metabolism. It is also known as hexose monophosphate pathway shunt. So the pentose phosphate pathway is an alternate metabolism to glucose, and it is also known as HMP shunt, and it is more complex pathway than glycolysis. Glycolysis, when you study, you might feel very easy because the steps are clear, but in HMP shunt pathway, the steps are very complicated. So it is a complicated pathway. HMP shunt is more anabolic in nature, and HMP shunt helps in three ways. First, for the formation of NADPH, which will help in the synthesis of fatty acids and steroids, and it is responsible for maintaining the reduced glutathione for antioxidant activity, and it is also very essential for the synthesis of nucleotide and nucleic acid formation. In the exam, question might come: how the HMP shunt helps in human being? Or question comes like significance of HMP shunt. Then you have to write these 3 points. About 10% of glucose entering in this pathway every day. So around 10% of glucose in the metabolism enter inside HMP shunt pathway or PPB, pentose phosphate pathway. The liver and RBC metabolize about 30% of glucose by this method. The remaining methods, glycolysis, PCA cycle. In this, 30% of the glucose from erythrocyte and liver comes through this pathway only. So the HMP shunt takes place in the following tissues like adipose tissue, adrenal gland, erythrocytes, testis, lactating mammary gland. So these are the tissues which are highly active for HMP shunt. Most of these tissues are involved in the biosynthesis of fatty acids and steroids. That's why these tissues are highly active for HMP shunt pathway, because from these tissues only we are going to get the fatty acids and steroids. And for this synthesis, the synthesis of fatty acids and steroids needs NADPH molecule. The NADPH molecules are mostly produced through HMP shunt or PPP, phosphate or pentose phosphate pathway. So HMP shunt, this occurs in the cytosol of these tissues. Oxygenation is achieved by dehydrogenation using NADP and NAD plus molecule. The HMP shunt pathway is carried out in two steps. Step 1, the major steps, two major steps, two phases like glycolysis, energy investment phase and energy harvesting phase in glycolysis. HMP shunt, there are two phases. First one is irreversible oxidative phase, and second phase is reversible non-oxidative phase. Let's see the reversible oxidative phase. During this phase, three molecules of glucose 6 phosphate give rise to three molecules of carbon dioxide and three molecules of 5 carbon sugars. That is, 5 carbon sugars means pentose. So irreversible oxidation phase give rise to three molecules of carbon dioxide and three molecules of 5 carbon sugar from three molecules of glucose 6 phosphate. So the starting compound is glucose 6 phosphate, three molecules of glucose 6 phosphate. So in reversible non-oxidative phase, rearrangement to regenerate two molecules. That is, within the structure, within a chemical structure, the elements moves, that is rearrangement to regenerate two molecules of glucose 6 phosphate and one molecules of glyceraldehyde 3 phosphate. So this is what happening in the non-oxidative phase. HMP shunt is an unique multifunctional pathway. It starts with glucose 6 phosphate molecule, and during HMP shunt pathway, no ATP is directly utilized or produced in HMP shunt, whereas in citric acid cycle and in glycolysis, ATP molecules are produced, but in this pathway, no ATP molecules are directly utilized also and produced also. So it is a multifunctional pathway. Several interconvertible substances are produced in different directions in the metabolic reactions. So during the process of pathway, various interconvertible substances are produced in different directions. Let's see the steps one by one. Let's see the steps one by one. First step is oxidative phase. So in oxidative phase, we have various steps. Step 1. In step 1, glucose 6 phosphate is oxidized by NADP dependent glucose 6 phosphate dehydrogenase to form 6 phosphogluconolactone. So in the step 1, glucose 6 phosphate is converted into 6 phosphogluconolactone with the help of the enzyme glucose 6 phosphate dehydrogenase. This is an NADP dependent enzyme. NADPH is formed in this step, and this is a rate limiting step. Step 2, 6 phosphogluconolactone, which is produced from the step 1, is undergoing hydrolysis, and with the help of the enzyme gluconolactone hydrolase to form 6-phosphogluconate. So in second step, the 6-phosphogluconolactone is hydrolyzed by the enzyme gluconolactone hydrolase to form 6-phosphogluconate. Now let's see step 3. In step 3, the synthesis of NADPH occurs. So for this, catalyzed by the enzyme phosphogluconate dehydrogenase. Because of this enzyme activity, we are getting a substance called 3-keto 6-phosphogluconate. 3-keto 6-phosphogluconate. So from where we are getting this? We are getting this from 6-phosphogluconate. 6-phosphogluconate will be catalyzed by the enzyme 6-phosphogluconate dehydrogenase. 6-phosphogluconate is catalyzed by the enzyme 6-phosphogluconate dehydrogenase to produce 3-keto 6-phosphogluconate. 3-keto 6-phosphogluconate. This undergoes decarboxylation to form ribulose 5-phosphate. This substance 3-keto 6-phosphogluconate undergoes decarboxylation to form ribulose 5-phosphate. In step 4, the ribulose 5-phosphate is then isomerized to ribose 5-phosphate. Ribulose 5-phosphate undergoing isomerization in step 4 to form ribose 5-phosphate, or it undergoes epimerization to form cellulose 5-phosphate. In step 4, ribulose 5-phosphate, if it undergoes isomerization, it forms ribose 5-phosphate. If it undergoes epimerization, it forms cellulose 5-phosphate. Now let's see step 5. In step 5, the step 5 is a non-oxidative phase. The step 3, so first phase having 3 steps. From step 4, non-oxidative phase starts. So step 5 is non-oxidative phase, that is trans-ketolase reaction. Step 5 is a trans-ketolase reaction. Step 6 is trans-aldolase reaction. So what is trans-ketolase? Trans-ketolase substance is a thymine pyrophosphate dependent enzyme. Trans-ketolase, that is an enzyme that is made up of thymine pyrophosphate. So this enzyme needs thymine pyrophosphate. So in step 5, the 2 carbon unit from cellulose 5-phosphate. So in step 4, we have seen cellulose 5-phosphate. This cellulose 5-phosphate, that is 2 carbon unit from cellulose 5-phosphate, will be converted to ribose 5-phosphate. That is, transfer occurs between cellulose 5-phosphate and that between cellulose 5-phosphate and ribose 5-phosphate. So you can see in the step 4, ribose 5-phosphate we got, also we got cellulose 5-phosphate. So between ribose 5-phosphate and cellulose 5-phosphate, what is happening? Transfer of 2 carbon unit is occurring. As a result of this, so what is happening? Already we have pentose, that is 5-phosphate. So 5-phosphate is pentose. So that from that 2 phosphate molecules add, that gives so 2 carbon from the, that is pentose, which have 5 carbon unit. So from that 2 carbon units are adding, so that it forms a new substance which is having 7 sugar molecules. That is, 7 carbon molecules will be present. That substance is called pseudoheptulase. Also, we are getting another substance called glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. Now, let's see step 6. In step 6, that is trans-aldolase reaction. Trans-aldolase brings about transfer of 3 carbon fragment from pseudoheptulase to glyceraldehyde 3-phosphate. So in step 6, 3 carbon fragments are transferred from pseudoheptulase to glyceraldehyde 3-phosphate. As a result of this transfer, we are getting a new substance called fructose 6-phosphate and 4 carbon erythrose 4-phosphate, and we are getting a substance fructose 6-phosphate and 4 carbon molecule erythrose 4-phosphate. So this is happening in step 6. Finally, in step 7, that is second trans-ketolase reaction, it is another trans-ketolase reaction. Already one trans-ketolase reaction takes place in the fifth step. Now the second trans-ketolase reaction takes place in the 7th step. So because of this, 2 carbon unit is transferred from xylulose 5-phosphate to erythrose 4-phosphate. So here 2 carbon units transferred between cellulose 5-phosphate and erythrose 4-phosphate. Because of this, we are getting a substance called fructose 6-phosphate and glyceraldehyde 3-phosphate. This fructose 6-phosphate and glyceraldehyde 3-phosphate will further metabolize by glycolysis and TCA cycle. So now you remember what happens with the fructose 6-phosphate and glyceraldehyde 3-phosphate in this reaction. So this cycle will continue. So this is what happening in the non-oxidative phase. So now to summarize that, what are the significance of HMP shunt? So it is a unique in generating 2 important pentose and NADPH molecule. So what are the importances of pentoses? In HMP shunt, hexoses, six carbon molecule, that is contained the substance containing six carbon molecules, hexose, are converted into high carbon molecule substance, pentose. The most common pentose produced in the HMP shunt is ribose 5-phosphate. So this pentose molecule is a derivative which is very useful for the synthesis of nucleic acids like DNA and RNA substance. And finally, the importances of NADPH is NADPH is required for the biosynthesis of fatty acid and steroids. NADPH is used in the synthesis of certain amino acids involving the enzyme glutamate dehydrogenase. NADPH is needed for synthesis of some amino acids. NADPH is needed for the synthesis of fatty acid, steroids, and some amino acids. That amino acid must contain the enzymes glutamate dehydrogenase. Also, this NADPH acts like a free radical scavenging. In our body, the radicals are produced. So these radicals have to be removed from the body. For that, NADPH molecules acts like a scavengers. These free radicals, examples are superoxide and hydrogen peroxide, are continuously produced in the cells because of various metabolic reactions. So these substances like superoxide and hydrogen peroxide has to be removed for that NADPH molecule helpful. And another importance is these free radicals, if it is not removed from our body, that will destroy the DNA proteins. Also, fatty acids are going to be destroyed, and other biomolecules are destroyed in the cells because of these free radicals. These free radicals are inactivated by the enzyme systems which contains SOD, POD, and glutathione reductases. This SOD means superoxide dismutase. POD means peroxide. GSH means glutathione. The glutathione is regenerated. The reduced glutathione is regenerated again with the help of NADH. So these are the importances of NADPH. In the exam comes importances of NADPH. You can write these things. So finally, to summarize the steps, so first step, glucose 6-phosphate will be converted into 6-phosphogluconolactone, the enzyme responsible as glucose 6-phosphate dehydrogenase. So there NADP molecule is reduced to NADPH substance. So second, the hydrogenation occur, so 6-phosphogluconolactone is converted into this one 6-phosphogluconate with the help of the enzyme gluconolactonase. This 6-phosphogluconate is again converted into ribulose 5-phosphate with the help of the enzyme phosphogluconate dehydrogenase. So this is a non-oxidative reaction starts. So the first three steps comes under oxidative phase. A non-oxidative reaction starts here. So ribulose 5-phosphate undergoes epimerization. Also we have seen isomerization to give two different substances, cellulose 5-phosphate and ribose 5-phosphate. The cellulose 5-phosphate and ribose 5-phosphate undergoes first trans-ketolase reaction to form pseudoheptulase 7-phosphate and glyceraldehyde 3-phosphate. So that undergoes trans-aldolase reaction. We get erythrose 4-phosphate. Also we get fructose 6-phosphate. Now this cellulose 6-phosphate will again combine with erythrose 4-phosphate and to form second, that is seventh step, that is trans-ketolase reaction to give glyceraldehyde 3-phosphate and fructose 6-phosphate. So in the exam HMP shunt pathway, draw this diagram. Very easy to give marks by the examiner who is evaluating your paper. So that's all about HMP shunt pathway. Hope you enjoy this lecture. Thank you for watching. Meet you again with another important questions. And we are going to complete the carbohydrate unit in another one lecture. Thank you all. Take care. Bye bye.